Peptic ulcers and the drugs to treat them. Peptic ulcers are painful sores that form in the lining of the gastrointestinal tract. They occur in the stomach or upper intestine and less frequently in the lower esophagus. They affect up to 24 million people each year globally, impacting their lives through painful symptoms of abdominal pain, nausea, and bloating. The formation of these ulcers is highly dependent on the health of the gastrointestinal lining. The GI tract is lined by the mucosa, a specialized tissue essential for digestion, secretion, and protection. It consists of 3 layers: the epithelial layer, lamina propria, and muscular layer. The innermost layer, called the glandular epithelium, secretes large amounts of mucus, enzymes, and bicarbonate, making it crucial to not only digestion but also defense against fluctuations in pH. The 2 main causes of peptic ulcers are Helicobacter pylori infection and the long-term use of non-steroidal anti-inflammatory drugs. H. pylori infections result in the formation of a peptic ulcer by causing inflammation in the GI tract. H. pylori also produces ammonia, which confuses the glands into thinking that the stomach is more basic than it actually is. In response, the parietal cells within the stomach produce excess acid. The acid damages the lining of the stomach, causing the ulcer. Another cause of peptic ulcers is the long-term use of NSAIDs, such as ibuprofen.
Even though NSAIDs help reduce inflammation, pain, and fever within the body, people who take NSAIDs are at increased risk of developing peptic ulcers due to this decrease of prostaglandin activity, which plays a huge role in protecting the lining of the stomach. Prostaglandins stimulate the production of mucus and bicarbonate to keep the stomach protected from stomach acid, but with them inhibited, the lining becomes weaker, and thus peptic ulcers join the party. Misoprostol is a medicine used to combat pesky peptic ulcers in those who are taking NSAIDs to reduce their risk of developing an ulcer by protecting their stomach linings in advance. Taken orally in tablet form, it exists as a synthetic methyl ester analog of prostaglandin E1, which inhibits gastric acid and pepsin secretion, and thus protects the gastrointestinal mucosa from damage. Misoprostol both inhibits gastric acid secretion and enhances mucosal protection. When inhibiting, misoprostol inhibits proton pumps in epithelial cells in the stomach by blocking a CAMP-dependent pathway, resulting in a decrease in H+ ions within the gastric lumen, and thus gastric pH is increased. An increased pH means a decrease in stomach acidity, meaning the stomach lining is now more protected. The stomach lining is now happy. Misoprostol also promotes mucus and bicarbonate production, 2 natural components of the stomach lining. It binds to the EP3 receptor on epithelial cells, enhancing the production of bicarbonate and mucus, and thickening the mucosal layer by causing edema in the mucosa and submucosa, reducing the backflow of hydrogen ions and enhancing mucosal blood flow regulation. As a result, the mucosa's ability to regenerate new cells is preserved. The stomach lining is even happier now. Bye-bye, peptic ulcers!
Another treatment for peptic ulcers are proton pump inhibitors, or PPIs. PPIs are oral medications derived from bezinomyzole that reduce stomach acid production. Some common prescribed PPIs include omeprazole, lansoprazole, pantoprazole, and esomeprazole. You notice that all PPIs are denoted by the suffix prazole. So how do PPIs treat peptic ulcers? The mechanism of PPIs works by inhibiting the hydrogen potassium ATPase enzyme, also known as the proton pump, lining stomach parietal cells, whose role in forming hydrochloric acid is vital in stomach acid production. When a PPI diffuses into a stomach parietal cell, it is activated by acid-base cleavage in the acidic cell environment. The active PPI then covalently bonds to the proton pump, irreversibly inactivating it and inhibiting the transport of hydrogen to join chloride to form hydrochloric acid as a stomach acid component. This mechanism results in an overall reduction of gastric acid, which enables and supports the healing of peptic ulcers by adjusting the stomach pH to normal levels in other gastric diseases, including Helicobacter pylori infections. It's also important that stomach acid production is not completely inhibited. The amount is reduced for normal digestion, as only a portion of the proton pumps in the stomach are blocked. Research has found that cases of long-term use of PPIs can lead to increased risk of enteric bacterial infections, gastric polyps, and gastric and colorectal cancer.
Besides PPIs, antibiotics are another form of treatment for peptic ulcer disease. Specifically, it is primarily used for Helicobacter pylori-associated peptic ulcer disease. The first line of treatment is a triple therapy consisting of 2 antibiotics and a PPI. Amoxicillin, clarithromycin, and metronidazole are the most common antibiotics used. Amoxicillin is a beta-lactam antibiotic. It binds to the penicillin-binding proteins, which inhibits peptidoglycan synthesis and thus inhibits bacterial cell wall synthesis. This ultimately leads to cell death. Next, clarithromycin is a macrolide antibiotic that binds to the 23S bacterial ribosomal RNA. This prevents the translocation of amino-acyl transfer RNA, thus inhibiting bacterial protein synthesis. However, mutations within the 23S bacterial rRNA binding site decreases the binding affinity of clarithromycin, leading to reduced inhibition of bacterial growth. Lastly, metronidazole inhibits bacterial protein synthesis by disrupting the helical DNA structure and causing DNA strand breaks, which ultimately leads to bacterial death. Having said that, more than 80% of H. pylori are resistant to metronidazole. Hence, it is often prescribed in combination with one or more antibiotics. Point mutations in the H. pylori genome have driven increasing antibiotic resistance over time. To overcome this challenge, bismuth-containing quadruple therapy has been recommended as the revised first line of treatment, with bismuth acting as a barrier against increased mucosal protective factors produced by H. pylori.